Hello everyone, and thank you for joining this presentation. Today we'll be talking about a fundamental but often underestimated topic in research data management: how to organize your data effectively. Organizing your data is about making your research reproducible, shareable, and ultimately more impactful. Let's dive in and explore some practical strategies and tools to help you do just that. Our agenda consists of 6 points. Principles for handling research data, handling data tables, handling images, organizing and sharing data, write a README file, and finally, checklist for reproducible research. Let's dive in. Research data comes in many flavors and shapes: tables, images, videos, text documents, and more. No matter the type of data, it's essential that your dataset has a clear structure and is understandable to others. A good tip is to put yourself in the shoes of an outside observer, someone who is not familiar with your project. Would they be able to navigate your data and understand what each file means? Without a clear structure, even the best research can get lost in a sea of confusion. The first step to making your data understandable is to use clear and consistent naming conventions. A good file name should fairly describe the file's content and make it easy to relate it to other files. For example, instead of naming a file something vague like a 1.tiff, use a descriptive name containing analysis factors like experiment underscore, mouse underscore, date underscore, condition underscore, marker underscore .tiff. Or instead of cells table, XLS, use something more specific like widefield underscore 5x underscore cortex underscore noon underscore counts as CSV. This small effort makes a huge difference when you, or someone else, revisits the dataset later. The second key practice is to prioritize open and accessible file formats. Using proper formats improves the accessibility and longevity of your data, making sure that others, and your future self, can open and use the files without relying on specific software. For example, use .tiff files for images to preserve both quality and metadata. Store tables as .csv files instead of proprietary formats. For graphs, prefer PNG or SVG formats, which preserve image quality. And for documentation, save it as .txt or PDF to ensure broad compatibility. Choosing open formats is a simple but powerful way to make your data more future-proof. The third key element is to provide comprehensive metadata. Metadata helps to contextualize and describe your research files so that others can understand not just the data, but also how it was collected, processed, and structured. At a minimum, you should include README files and data dictionaries or codebooks. For example, as shown here, a good codebook clearly defines each variable, its description, type, and possible values. This documentation turns a raw dataset into an understandable and reusable resource. The fourth best practice is to implement reproducible workflows. Whenever possible, use coding tools like R or Python to transform your raw data into data ready for analysis. By using code instead of manual edits, you ensure that the process is transparent, repeatable, and much easier to validate. These practices help you produce datasets that are organized, clean, and validated, making your research more robust and trustworthy. Let's explore essential practices for handling data tables. Tables are the core of most scientific datasets. Even though spreadsheets, like Excel files, are the most common way to record and store data, they're often poorly organized and sometimes even unusable. Here, you can see some real examples: spreadsheets with merged cells, inconsistent formatting, or mixed types of data in the same column. These issues create major obstacles for analysis and reuse later. A well-structured table is essential for making your data clean, analyzable, and trustworthy. Unfortunately, journals accept poorly organized tables. Here are 2 examples from recent publications. In both cases, you can see issues like merged cells, mixed types of information in the same columns, and visual elements like color codes that are not properly documented in the data. These practices make the data harder to analyze, reuse, or even understand. Good data organization is not just a technical detail. It's a fundamental part of scientific quality. Let's look at more examples, this time from Crystal Lewis's work on data organization. In these comparisons, you can see exactly what separates bad data practices from good ones. On the left, you see common problems from top left to bottom right. Irregular structures, numeric data masking as text, inconsistent formats, and combining multiple pieces of information into a single column. On the right, you see the corrected versions, top left to bottom right. Rectangular tables, numeric data stored as numbers, standardized formats, and one clear variable per column. Following these best practices makes data easier to analyze and share, and much more reliable for future research. Here are two more important lessons from Crystal Lewis's work. First, data should always be explicit. In the table on the left, repeated values like school IDs and years are omitted, assuming the reader can infer them. But this creates confusion and errors during data processing. On the right, the corrected table repeats these values explicitly for every row, making the dataset complete and machine-readable. Second, avoid relying on visual cues like color to convey information. In the left table, treatment groups are indicated by cell color, a method that computers cannot easily interpret. The corrected version adds an explicit treatment column with clear numerical values, ensuring that all critical information is captured directly in the data itself. Now let's look at how to build accessible and well-structured data tables. Here, we have a clean and organized example. Each column plays a clear role. Identifier variables like animal ID, time point, and condition describe the grouping factors. Analysis variables include the actual data you measure, like scores, areas, or cell counts. We can also have variables created during processing, such as proportions or ratios. Note that each row contains the values for a unique observation, making the dataset tidy and easy to analyze. Now let's talk about table formats, starting with wide format tables. In a wide format, each subject occupies a single row, and each variable is placed in its own column. You might have columns for subject IDs, different variables like var1 and var2, or repeated measurements at different times, such as time 1, time 2, and time 3. This structure is often used when preparing data for regression models, where each column can serve as a response or predictor. For example, you might model cells underscore 7D as a function of cells underscore 2D and cells underscore 3D values collected across different time points. Now let's look at long format data tables. In a long format table, each subject appears multiple times, once for each observation or time point. Instead of having different measurements spread across multiple columns, the observations are organized into rows, with an additional column to indicate the time point or condition. This structure is especially useful when working with time-lapse data or repeated measures, where you want to group observations efficiently. For example, you might model cell counts as a function of the time point, 1 day, 2 days, or 3 days after an intervention. Long format is usually the preferred structure for statistical analysis because it makes modeling much more straightforward. And the best part is that you don't have to choose forever between long or wide formats. Using tools like R or Python and platforms like Cordo, you can easily reshape your data. For example, in R, you can use pivot_wider to go from long format to wide format and pivot_longer to do the reverse. This flexibility allows you to adapt your data structure depending on your analysis needs, without losing any information. There are excellent tutorials available if you want to practice these transformations. Another critical step in making your data understandable and reusable is to provide descriptive metadata. Datasets without metadata, like codebooks, data dictionaries, or README files, are often unintelligible to others. At a minimum, you should include a README file usually in plain text or markdown format. It should explain the context of the project, describe the content of each file or folder, and define the variables contained in your data tables. Here's an example showing how clear documentation can guide users through a dataset, ensuring they understand where the data came from, how it was processed, and how to use it properly. Now, let's explore essential elements for handling images. When it comes to handling images, there are a few important best practices to keep in mind. First, always convert proprietary formats, like .xi files, into open, non-compressed formats such as .tiff. This ensures your images can be opened and preserved in the long term. Second, share both technical metadata, like acquisition parameters, and descriptive metadata that explains the context and content of the images. Third, document all image processing steps using coding or scripting tools. For example, resizing, background subtraction, or other transformations. Finally, perform your analyses using coding software whenever possible, and avoid manual, non-reproducible workflows. If you want to dive deeper into these practices, we recommend visiting the resource linked here. When working with microscopy images, it's important to transform proprietary formats like .xi into open formats like .tiff. One easy way to do this is by using Fiji, a free image processing tool. With simple scripts like the one shown here, you can automate the conversion process, selecting a folder, opening each .xi file, and saving the images in .tiff format. However, be aware when saving .xi images as .tiff, you will lose some embedded metadata stored inside the original file. That's why it's critical to separately archive important acquisition information when transforming files. When working with research images, it's crucial to keep track of two types of metadata: technical and descriptive. Technical metadata includes acquisition details like pixel dimensions, number of channels, and image depth. In Fiji, you can easily export this information from proprietary files like .xi into .txt or .csv files, and even batch export it from multiple images. On the other side, descriptive metadata provides the story behind the data, explaining the provenance, the experimental context, and the naming conventions used in your dataset. Both types of metadata are essential to make sure your images can be properly interpreted, reused, and cited in the future. It's time to revisit the basic principles for organizing data. These same principles apply to data that is intended to be shared with the public. We are currently living through a concerning era in research, a pandemic of fraudulent and irreproducible science. As this graph shows, the number of retracted scientific articles has been rising sharply over the past few decades, particularly in biomedical sciences. This worrying landscape reminds us that we, as responsible researchers, must commit to good scientific practices. A well-structured dataset is the key to making your research understandable, reusable, and reproducible. Think of it like a set of matryoshka dolls, each part neatly organized within the next. On the right, you see an example of a good file structure for a research project. Raw data is separated from processed data. Scripts are organized in a code folder, and outputs like figures and models are saved systematically. There's also a README file to document the entire structure. This approach not only makes collaboration easier, but also ensures that future users, including your future self, can navigate and understand your dataset. When it comes to structuring your dataset, the ideal time to define your organization is right at the beginning of your project. If that's not possible, setting up a clear structure during your research is the next best thing. There are a few key things to think about. First, plan your folder and directory structures carefully. Second, think ahead about the file types and formats you'll use. Third, establish logical and descriptive naming conventions for your files and folders. Overall, your dataset should be logical, consistent, and easily understandable by external users, often people unfamiliar with your project. One excellent way to organize your research data is by following a standardized folder structure. The Tier 4.0 protocol provides a ready-to-use project template designed to bring consistency and transparency to datasets. You can download the Tier 4.0 project structure and adapt it to your specific research needs. As shown here, the template organizes everything systematically, from raw data to processing scripts to final outputs and reports. The data_off folder is where you should store your original, unprocessed files. This can include original images, such as .tiff or .xi files. Also, output files directly from measurement devices, like .txt or .csv, or original registration datasheets in formats like .png, .csv, or .xlsx. Keeping your raw data separate and organized is essential for verification, reprocessing, or reanalysis. In addition to storing your raw data, it's essential to include metadata that allows others, and your future self, to understand and reuse the files properly. You should include methodological and technical details that describe how the data was generated. Also, provide codebooks or data dictionaries that explain the variables, units, and structure of your datasets. These can be shared as simple .txt files or in spreadsheet formats like .csv. And finally, for images, remember that saving and sharing the instrument settings and acquisition parameters is critical for correct interpretation. After raw data, the next important element is your process data. The data_analysis folder contains the files that have been cleaned, transformed, and prepared to generate your research results. Just like with raw data, you should include metadata to explain the process files clearly. It's also helpful to include data appendix files, basic tables or reports that show descriptive statistics, data distributions, or any transformations applied. Sometimes, your analysis pipeline includes additional steps between raw and final process data. In that case, it's helpful to create a data intermediate folder. This optional folder can hold intermediate files such as image masks, filtered datasets, or even machine learning classifiers that are used in further processing. While many scientists are still more comfortable using graphical interfaces, the reality of today's research environment is clear. Scripting and coding are essential tools. Using code in R, Python, or any open-source environment ensures that your analysis is transparent, trackable, and reproducible. Ultimately, coding should be treated like any other core research method. It's not just for computer scientists. It's for every researcher who values integrity and reproducibility. Fortunately, there are excellent tools available to help researchers write, document, and share their code. On the left, you see RStudio paired with Quarto, a powerful combination that lets you work in R or Python, create dynamic documents, and integrate code, text, and outputs all in one place. On the right, GitHub is a version control platform that helps track changes, collaborate with others, and publish your code alongside your data. These tools are open, widely used in the scientific community, and essential for reproducible research workflows. With RStudio and Quarto, you can do a lot more than just write code. You can manage your entire research workflow in one place. You can handle data tables and variables efficiently using the R's tidyverse, which simplifies data cleaning and transformation. You can analyze images using Python libraries like skimage and process flow cytometry data with our packages like Flowcore from Bioconductor. For gene expression studies, tools like DSeq2 make RNA-seq analysis reproducible and scalable. And when it comes to statistical modeling, packages like BRMS let you build powerful models with clear, transparent code. To manage your code and data effectively, version control is your best friend. Platforms like GitHub and GitLab allow you to securely store your code, collaborate with others, and make your work publicly available if desired. You can track the entire history of your project and create official versions like 1.0, 1.2, or 2.0. These platforms also integrate with repositories like the Open Science Framework, letting you link and render your code directly alongside your research outputs. And perhaps most importantly, by sharing your work openly, you support other researchers and contribute to a culture of transparency and reproducibility. One of the great things about learning to code today is that you don't have to do it alone. There are amazing global communities that support diversity and inclusion in programming. Our Ladies is an international organization that promotes gender diversity in their community, offering mentorship, workshops, and a welcoming space to learn and grow. PY Ladies provides the same kind of support for Python users, building a global network that empowers women and gender minorities in data science and software development. The scripts processing folder is where you keep the code that prepares your raw data for analysis. These scripts might drop unnecessary variables, generate new ones by performing computations, or merge multiple data sources into a unified dataset. This part of your workflow is critical as it defines how raw data becomes structured, analyzable information. And as we've seen before, it's a good practice to save the outputs from these steps, your intermediate files, in a dedicated data intermediate folder to keep everything organized and reproducible. Keep in mind that logical naming conventions are the key to linking the raw data, processing scripts, and analysis data. The scripts analysis folder holds the code used to generate your research results. This includes scripts that produce images, figures, tables, or statistical models. It's the final step in your pipeline, where the analysis comes together and becomes publishable results. The scripts folder can also contain a master script that executes all other scripts, creating a fully automated pipeline. The results folder is where you store the outputs generated by your analysis scripts. This can include static or dynamic visualizations, such as images and figures, as well as tables and statistical model summaries. Each of these results should be traceable, meaning it's clear which script and dataset produced them. A well-organized results folder not only helps with writing your publication, but also ensures that others can validate and build on your work. Let's now talk about README files. README files are one of the most important tools for data understanding and reuse. They serve as guides to help others interpret your dataset and tables. A good README provides context, describes file contents, and explains how to use the data. There are many resources to help you create a clear and effective README, like readme.so and readme.ai, among others. A well-crafted README file is your dataset's user manual, and it should stand on its own, without requiring readers to refer to the related article. Here are the key elements to include. First, provide a dataset identifier. This includes the title, author names, data collection dates, and any relevant geographic information. Next, include a map of files and folders, showing the organization of your dataset, along with naming conventions. Also include methodological details, such as how the data was collected, processed, and under what experimental conditions. Don't forget the instructions and required software to open or reproduce your analysis pipeline. Finally, specify the sharing and access conditions so users know how they are allowed to reuse the dataset. And one important reminder, your README must contain the methodological context. It should never rely on a separate article to explain how the data was created. Finally, let us review the essential elements that a dataset should have in line with open and reproducible research practices. A reproducible research project is well-documented, well-structured, and transparent. It starts with a clear folder and file organization, using open file formats like CSV or TIFF and consistent naming conventions. This makes your data navigable and understandable. Next, your workflows from data cleaning to analysis should be reproducible. This means using scripts that allow anyone to recreate your results, images, tables, models, or plots. And finally, a good project includes README files and data dictionaries that provide full context. What the dataset is, how it was created, what variables it includes, and how to interpret them. Reproducibility is a mindset and a commitment that builds trust and drives science forward. To summarize, your dataset is not just supporting material. It's an independent research object. It can be shared, reused, and cited on its own, independent of the article it may be associated with. In fact, it's time we flip the perspective. The article must become a supplement to the dataset, not the other way around. If you're looking for guidance on how to properly deposit your data in trusted repositories, visit the linked resource. To close, here are some key resources to help you put these principles into action. You'll find detailed guidance in the FRDR documentation, hands-on training materials from the Alliance, and practical tools in the RDEEM kit developed by Elixir Europe. This presentation is also available online in both English and French by scanning the QR code or visiting the GitHub link shown here. And if you need support at any stage, don't hesitate to reach out. Our team is here to help you prepare, document, and share your data effectively with the research community. You can contact us by email at rdm-gdr@alliancecan.ca or visit our website to learn more.